All right, welcome to week 10 on variation genetic crosses. Your keywords, chromosomes, gamete, and gene. There's going to be a lot of keywords in this lesson. So just a couple there to start with. Your core knowledge questions, there are 3, and your challenge at the end there. So have a read, have a go, and then we'll come back and see how you did. So let's see how you did. Give the energy transfers an electric fan. Identify the useful and the wasted. So if we plug an electric fan in, it's going to be electrical going in. And then your kinetic to move the fan blade. That's going to be the useful energy. And then if we move over, you're going to be able to hear the fan. So that's a wasted energy because there's no benefit to that. And then if you leave it on long enough, it's going to start warming up slightly. Heat is wasted energy there. Draw a dot and cross diagram for the electron configuration for lithium. So we've got a mass number of 7, a proton number of 3. So the proton number is what we need because it's always the same number of protons as electrons. So I've done my pen there. So 3 protons, 3 electrons. There's 4 neutrons, 7 minus 3 there. Big number minus a small number is going to give you a neutron number. There's my electron configuration, my diagram. So I got 2 in the inner shell and I've got 1 on the outer shell there. My inner shell can only take 2 electrons. My electron configuration is 2, 1. Just a second, love. And then I've got my challenge question. A kettle uses 3 kilowatts. Well, a kilowatt is 1,000 watts, same as a kilometer is 1,000 meters. So I've just broken that down to 3,000 watts of electricity to heat water. 300 watts is lost as sound, which is not very useful. It's the heat energy that's useful. What is the efficiency of the kettle? So I've said there's 3,000 going in. Remember the conservation of energy. Energy going... Go back in the room a second, lad. Energy cannot be created or destroyed. It can only be transferred. So the energy going in must equal the energy coming out. So I've got 3,000 going in. So I need 3,000 watts coming out. And I've got 300 as sound. So therefore, 300 plus something must equal 3,000. And that something is 2,700. So that's my useful energy, heat. Now that I know my useful and my total, I can use this equation. Percentage equals useful over total times 100 to give me my efficiency. So that's 2,700 over 300 times 100 is going to give me 90% efficient. It's probably not going to be that efficient, a kettle, but these numbers I've made up. So today's lesson is on predicting genetic outcomes using genetic crosses. So we're going to use these crosses. Now, your keywords, chromosome, gamete, gene, but we're going to be using a lot more words that you've probably never heard before. This is linked with your GCSE biology. There's kind of 2 parts. Key stage 3 are going to be expected to do one thing. Key stage 4 are going to be expected to do another. But it all kind of ties in. And I would be expecting your key stage 3s to have a bit of a go anyway, because we do in normal lessons, so there's no reason we wouldn't today. It's just a lot of terminology, a lot of stuff to get your head around. I'm not a biology teacher. I'm going to do my best to break this down. It's a topic that I've struggled with in the past, especially because of all the key terms. So we're going to be dealing with 4 key terms just to begin with before we have a little bit of a quiz. And the first one is gamete. Now there's a bit of information you can read there, but basically a gamete is a sex cell. You can see Miss Gundy about what happens. I'm not going to go into the details of it, but basically men have sperm and females have eggs, and that's pretty much it. You get half your genetics from your dad, half your genetics from your mother. So these sex cells, or these gametes, carry half of the information. So whether you've got blue eyes or brown eyes is determined really by the combination that's come from these gametes that's been passed on. So that's what a gamete is. There's a little bit about DNA there. We're going to be mentioning DNA too much because DNA and the next word, chromosome, are used kind of interchangeably here. It's the same sort of thing. So first thing you need to know, gamete is a sex cell. It's either a sperm cell or an egg cell, and it carries half of the genetic information. What's the next one? Chromosome. I just mentioned that word, chromosome. Kind of like, well, it is DNA. Chromosome are just strands of DNA. So if I show you these Lego blocks, you get one from your mum and one from your dad, chromosomes, and they come together. And humans have 23 pairs. So we've got 43 of these Lego blocks in total. Okay, one from your mum, one from your dad, passed along. So 23 pairs, and these will code for different things like hair colour, eye colour, the shape of your ears, the shape of your nose, how high you are, how broad you are, many different things, skin colour, you know, if your beard's going to be ginger or not. It's all determined by your chromosomes. These things, you get 23 pairs, so 46 in total, okay? And there's just a picture of that there. You can see that this is the diagram for the genetic code, really, the chromosomes. And obviously we're dealing with humans here. Many animals got many more. Your next key term, so we talked about your gamete is your sex cell that carries these things in the nucleus. Okay, all genetic codes carried in the nucleus. Then you got your chromosomes, and then you got genes. And genes, if we look at this Lego block, if you see I've got 2 blocks of blue, and then I got 1 block of grey, and then I've got 2 blocks of blue, and then I got a yellow and an orange there. Now they're the same gene, but we're going to be talking about this now. So these are genes. These code for particular things. The shape, whether you've got a gap in your teeth, whether you can whistle, whether you can roll your tongue, are all determined by genes, and you've got loads of these, okay? So don't get fooled in thinking you've only got like 4 genes. I'm just using the Lego to represent it. So this one's from my mum, from my dad. It doesn't matter really which ones I get. But you can see here that, you know, I got matching genes, okay? And it says there's small sections of DNA. I didn't want to do that there. There's small sections of DNA. So they're just chunks of DNA. These amino acids, these proteins that make up your characteristics, okay? And in each cell we carry these, except for red blood cells and a couple of others, okay? There's always going to be that genetic code to tell your body what to do, how to make it. Chromosomes in each pair is carried in the same location. The genes can be different or the same. Now this next word is the word I struggled with for ages, and it's called an allele. Where's it to? It's down by here. The allele, which is pretty much the same as saying a gene. So remember now, you've got your gamete, your sperm coming in, it's carrying half, the egg, okay? And they meet, okay? Once the sperm goes inside the egg, they meet, and here's just a little bit of a chromosome, okay? One piece of chromosome here that carries genes. These genes, I've got a grey one, an orange one, a red one, and a green one there. Now, an allele is a different version of the same gene. Oh, what does that mean? Different version of the same gene. If you can look at the grey one, they're the same gene there. So maybe that codes for the shape of your nose, okay? I got this one from my mum, this one from my dad, and it doesn't matter which one my body chooses, essentially, or which one is dominant. We'll talk about that, because I'm going to get a grey, I'm going to get this shaped nose no matter what, because they're identical genes. But sometimes there's a gene that's not the same. They still code for the same thing. So let's just use eye colour for an example, and let's use orange and yellow for the colours of eyes. I know it's ridiculous. So my dad may have orange eyes. My mother may have yellow eyes. Okay, or I shouldn't say that. They could have different color eyes completely. Let's just say I get the genes for yellow eyes from my mother and the genes for orange eyes from my father. Now, if orange is dominant over yellow, I'm going to get orange eyes, and we're going to talk about that a little bit later. But really, I wanted to see what the term allele means. It's the same gene, but they're different. Okay. And I could talk about this gene. They're in the same location. It's the same gene. This could be the shape of ears. You can see I've got 2 different shapes there. And I'm going to display one of them. That's called your phenotype. And we're going to talk about that again. But I'm going to display one of these characteristics. So an allele is the same version, different version of the same gene. The same gene. So I've got gametes, chromosomes, genes, and then alleles, different versions of the same gene. Let's have a look how we did there. Okay, so test yourself, press pause, quick true or false, and let's go for it. Lovely. Let's see how you did. Another term for a sperm or an egg cell is a gamete. That's right, that's the science term we use for sex cells, so your sperm or your egg cell. We get our genetic information from DNA. We do. Okay, DNA is just like this big code of information that we use. Chromosomes are found outside the nucleus. No, they're found inside the nucleus. So your genetic information is in the nucleus. It's found in, that's the job of the nucleus, really. Genes are made up of many chromosomes. This was a tough one, but it's actually the other way around. Chromosomes are made up of many genes. So this one is actually false. Oh, sorry, this one. Genes are made up of many chromosomes. It's actually chromosomes are made up of many genes. Both chromosomes come from only one parent. That's not true because we got two parents, so they must be half from each. So that's false. A gene is a slice of DNA. It is, yeah. A gene is a slice of chromosomes, which is DNA. Remember I said we use these words interchangeably, especially for this topic. Genes from either parent are usually identical. That's false. Sometimes they're identical. Sometimes you get two grey ones or two green ones, but usually they're not identical. That's why we've got that term alleles, different versions of the same gene. Different versions of the same gene are called proteins. Well, I just said no, they're not. They're called alleles, so they're not proteins, so that's false. If you're struggling with that, go back and have a look at the video again. Also, I would encourage you to look at other videos, okay? Actually go out and just search the term genetics, or Punnett Squares is what we're doing, genetic crosses. Because like I said, I'm not a biology teacher. I'm going to do the best I can, because this is a topic that I really struggle with, especially A-level and university. It got quite tough. Okay, two more words here, genotype, phenotype. I'm not going to dwell on this too much because once we start practicing, these terms get a little bit more familiar, if you know what I mean. So it's like learning a language. If I told you this means that and not put it into practice, it's really hard to access it in your brain. So a genotype is a collection of alleles. So a genotype is basically the letters that scientists use to represent, because we write letters. So it could be a capital A or a lower a, depending on which letter I write. Again, we need to go into practice this. So genotype, the letters. The phenotype is what's displayed. So if I say I've got, it's a 50% chance of me having green eyes, then that's the phenotype. It's the characteristic. The phenotype is a visible characteristic. Brown hair, blonde hair, I'm talking about a phenotype. But then when we're talking about letters, which we will do, that's your genotype, your gene. Okay, let's push on. Then we got dominant versus recessive. Dominant is probably a word you're familiar with. If you're dominant over something, it means that you're the one that's going to have your way. Okay, if I'm dominant in an argument, then it means that I'm winning that argument. Recessive is the opposite to dominant. So a dominant allele is represented by a capital letter, this comes back to your genotype. Remember, your genotype is letters. If it's a dominant, it's going to be a capital something, okay? Capital A or a capital E or a capital letter that either the examiner chooses or we choose. And your recessive allele is going to be a lowercase letter. Now, your dominant allele is always going to be displayed in the phenotype. The phenotype is the thing that's the characteristic that's displayed. See what I mean? It kind of gets complicated, but I'll tell you this now, the actual assignment that I set, the genetic crosses, are far easier to do than understanding the understanding behind it. I know that's a bit crazy. So key stage 3, you are able to keep up with this. We just need to get through this bit first. So your dominant is a capital letter. Your recessive is a lowercase letter. Dominant, if a dominant's present, if you see a capital letter, it's always going to be that color. For example, if I've got my chromosomes here and I've got genes on them, if this yellow from my dad passed on to me is dominant over orange, okay, even though I carry these two genes, the yellow is going to overtake. The orange is just not going to even come into effect. Let's go back to the example of yellow eyes and orange eyes, even though nobody's got yellow orange eyes. If I've got this gene for yellow eyes, if I've got this genotype, this capital whatever it is, it's always going to be displayed. Now, if I've got two orange ones, two of these recessive, then I will have orange eyes because orange is a recessive one. But I need two oranges to give me orange eyes. If I've got two yellows, then it doesn't matter because I've got two dominant ones anyway. I'm going to use either or. But if I've got a yellow and an orange, and yellow is dominant over orange, I'm going to have yellow. Okay? All right. Here's a good example with these. I think these are flamingos. Correct me if I'm wrong in the comments. But I've got an allele here, an allele, just a different version of the same gene. Okay, that's purple. This is pink. Okay, now which one is dominant over which? Well, you can see that this one brings purple to the table. This one brings pink. They mate, and the offspring has pink fur or pink skin. Pink is dominant, therefore, over purple. Okay, so pink is the dominant allele in this case. Last one, I promise now, homozygous, heterozygous. The term homo means the same. The term hetero means different. So homozygous alleles are the same. So if we look at my chromosomes here, oops, broke my Lego there. These two grey ones are homozygous. They're the same gene, okay? They're alleles, but they're the same. They're the same colour. So whether they're dominant or recessive, okay, they are homozygous. Now, you can get homozygous dominant, you can get homozygous recessive. If they're different, they're heterozygous. Now, there's no such thing as heterozygous dominant or heterozygous recessive because the dominant allele will always take it anyway, like I said about the yellow one. So even though they're heterozygous, the yellow one will take it if it's dominant, if that makes sense. Okay, bear with me. You can see my little diagram here. I've got homies there. They both got sunglasses. This one hasn't, so that's hetero. So what I want you to pick up from this really is homo is the same, hetero is different. You can see in this diagram here, homozygous, there's two sort of pink genes there, and there's different color genes there. Okay? It's easy to represent when you use a color, but these genes could represent anything. How hairy your chest is, how hairy your armpit is. These genes, they code for different characteristics in your physical makeup. And then we've got 4 combinations. When we start doing these genetic crosses, there's only 4 combinations you can come up with, really. And I'm just going to label these up. So homozygous, 2 capital B's. Because there's 2 capital B's, they're the same. So we call it homozygous, and this is going to be dominant. Homozygous dominant, because they're capital B's. Okay, they're the yellow gene. You can imagine how I got 2 yellows. So swap this orange for a yellow, and that's my homozygous dominant. They're the same dominant. I've got these 2 heterozygous then. I've got this big B and a little b. So if the yellow was big B and the orange was little b, that would be a perfect, that combination, big B, little b. Swap these then, get the other one from, so get this from your mum and this from your dad, and then it's going to be little b, big B, which you can also write big B, little b. We always put the capital letter first. So essentially they're the same. Can you see how this combination here, they're different, but they land up in the same sort of heterozygous, they're different heterozygous. And then the last combination really is homozygous, 2 little b's, but they're recessive this time. So this is homozygous recessive. Homozygous recessive. Homozygous, the letters are the same. Not the same in terms of, don't get a big misconception here is thinking you can have a B with a T. Okay, that does happen in other Punnett Squares, and usually in A-level. I forget the name. I think it's like second. No, it's not second generation. I think it's dihybrid. Dihybrid crosses, maybe. I don't know. Like I said, I'm not a biologist, but in this video, we're only going to be dealing with 1 gene. Okay, 1 allele. So it's going to be 1 letter, and it's either going to be capital or it's going to be lowercase. If they both match, if they're both lowercase, they're homozygous. If they're both uppercase, they're homozygous, dominant. If one's uppercase, one's lowercase, they're hetero, different, heterozygous. Let's put it into practice. Okay, well, test yourself first. Okay, press pause. See how you do this. The challenge, have a go. Brilliant. So let's see how you did. Heterozygous or homozygous. So these first two letters are different, therefore they're heterozygous, capital B, lowercase b. These two letters are both uppercase, so it's homozygous. And this would be homozygous dominant, okay, because they're both capital letters, dominant. This one then, lowercase big, capital, heterozygous again. This one, homozygous, because they're the same, both little e's, and this will be homozygous recessive this time. And then capital W, lowercase w, heterozygous. Just a quick note, it's very rare that we use these W's or C's or any letter that you can think that it's hard to differentiate between capital C and little c, okay? We tend to use capitals like T and R, that they look totally different, uppercase and lowercase. I would recommend that, especially when you have to choose the letter. Challenge you, if capital A represents brown hair and lowercase a represents blonde hair, what colour hair do the following have? Do you remember me saying about if the yellow is there, it's always going to be displayed? Okay, if yellow is dominant over orange, then that's it. Well, if a capital letter is there, it's always going to be displayed. And if capital A represents brown, then if capital A is there, it's going to be brown. It's going to have brown hair. Again, capital A is there. Even there's two of them this time. You're going to have brown. It doesn't mean it's going to be browner. It just means that you can only use one of these genes, see? Even though you got two, you can only use one and use the dominant one. Okay? And so you just have brown hair. It's the same, exactly the same brown hair as this. In this case, I got two lowercase. So I haven't got a dominant gene for brown. Therefore, my phenotype, what's displayed, is going to be blonde this time because I've got two recessive. I can only use one of these genes. Therefore, I must use one of the lowercase ones, which is going to be blonde hair. And this one's going to be brown hair because I've got that capital A. We could even go into a ratio here. I could say, or a percentage, that there's 75% chance of these having brown hair and 25% chance of having blonde hair if I was to take these four into account. And we're going to look at that now in these Punnett squares. So the genetic cross, I grabbed this from BBC Bite Size. I don't think it's the best one. I think you probably get more from the crosses when we start doing them. In fact, yeah, I'll go through it. So genetic cross is a single gene combination. I say a single gene because you can't get those dihybrid crosses where you start dealing with two genes at a time. But for GCSE, it's usually just one. And they can be examined using a Punnett square. This shows possible offspring combinations. And then you can calculate the probability, or you can calculate the ratio, depends which one you want. And it's easy maths because you're only dealing with 4 here. Remember I say there's only 4 possible combinations. So you're going to be dealing with easy numbers like 25%, 50%, or half, quarter, things like that. So we got a rabbit with black fur and a rabbit with black fur. Okay, so they're carrying 2 genes. Yeah, they're carrying a dominant for black fur and they're carrying a recessive. They're carrying a dominant and they're carrying a recessive. Now, even though you display the dominant, you can still carry the recessive. So there is a chance that 2 rabbits with black fur could mate, and there's a 25% chance there, or a 1 out of 4, quarter, that the rabbit could land up with white fur. Why is that? Because they both carry, this fella and this lady here, they carry the small, or the recessive gene. So when we cross these now, capital F is going to give us 2 capital Fs. So that's your homozygous because they're the same dominant. Then you're going to have capital F, little f. That's heterozygous because they're both different letters. Again, little f, capital F there, heterozygous. And then you've got little f, little f. So when you've got little f, and that big F is not there, then you will display, in your phenotype, you will display that recessive characteristic, which is sometimes blue eyes, sometimes blonde hair. Okay, the majority of us have got brown hair and brown eyes. So it's quite rare. I wouldn't say rare. Well, it's a 25% chance usually that you'll get blue eyes or blonde hair. We're just going to do these problems now together. I mean, I'm giving you the cross this time. So I'm giving you this thing. It's called the genetic cross. It's kind of like a noughts and crosses thing. Just want to go through some key terms before we dive into this. So remember now, a chromosome carries, you get one from each parent and it carries the genetic information. On these chromosomes, you've got genes, which is a slice of a chromosome. So you can actually take that gene out. Okay, I could take that gene out, which scientists have begun to do. There's a lot of controversy with that, when you can actually gene splice and take the genes out. That's another topic. Then you've got alleles, different versions of the same gene, orange, yellow. Yes, love? Oh, that's brilliant. Excellent. I'll be in now, I promise. Okay? Different version of the same gene. Then you've got your phenotype. What's displayed if I've got a ginger beard or a brown beard. I like a brown beard, but I've got a ginger beard, unfortunately. My genotype, the letters used for that, so you've got a capital letter or lowercase letter. It's going to be dominant or recessive. There we are, capital or lower. And then it's going to be hetero or homo, the same or different. Just to recap there. Problem 1. I'm going to do this one for you. The height of a pea plant. Now, biology's very, very interesting. It's very, very exciting when we're dealing with pea plants. All right, that's when you know you're there. Okay, the height of a pea plant is controlled by a single gene. Now you'll always have that in a question. They'll always try and complicate it. A single gene. They just mean, look, we're just dealing with one gene. We're going to keep things easy. Okay, and it's got 2 alleles. So it's got one there and one there. It's got tall and short. The tall allele is dominant, and it's got a capital T. So it's going to be tall if that capital T is there. It's going to be tall. But if the lowercase t, if you get 2 lowercase t's, it's going to be short. So that's the recessive gene. Complete the Punnett square to show different part combinations produced by the pea plants when they're bred. And this, in my opinion, is the easy bit. Even if you didn't know any of that before, there's a problem-solving process to this. So what you do, you match each one up. So it's called a genetic cross because you are pretty much crossing them, okay? You're crossing each one. So I'll rub that out now. Boop, 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 boop. So I got big T, little t. For this box here, okay, I got big T, little t. So big T, little t. Now I'm going to do this box. I've got big T, little t again, big T, little t. Can you see what I'm doing? I'm taking a letter from each of these boxes and I'm crossing them, a genetic cross. Now I've got little t, big T, and now I've got little t, big T. Hopefully you saw that. Hopefully you get that. I'll just do that again. I'll rub that out. Okay, I'll rub it out. And I'll do it again, just in case you didn't get it. Big T, little t. Big T, little t. Little t, big T. Little t, big T. Okay. Now it says there you should always write the dominant allele first, but for now, what that means, I should always put the capital letter first, but for now, you're not going to get marked down on this. And I'll just rewrite it. Big T, little t there. Big T, little t there. Now, how many tall ones am I going to get? Remember now, if a dominant allele is displayed, if it's there, it's going to be tall. I've got a capital letter here, I've got a capital letter here, I've got a capital letter there, I've got a capital letter there. I've got a 4/4 chance, which is 100%. Chance that it's going to be tall. Okay, because in order to get short, I'd need little t, little t. Let's do another one together. The colour of rabbit's fur is controlled by a single gene. Again, there it is. Grey and white. The grey allele is dominant and is capital G. The white allele is recessive and is a lowercase g. The female is heterozygous, which means it's going to have one big one, one little one. And the male is homozygous dominant, which means it's going to have two big ones. Homozygous, the same, dominant, big. Now, this one's already kind of done for you, and at key stage 3, this is what it'll look like. And already you have these, you've just got to cross them. So big G with little g, big G with big G, GG, big G with big G. I'll do that for you now. Hopefully this is the easy bit, big G, big G. Again, if you're wondering where I'm getting this from, I'm getting one from this box, one from this box. And then for this box here, I'm going to get one from this box because it comes over, and one from this box. So it's going to be big G, little g. Again, I'm going to go big G, big G, big G, big G, little g, big G, but I'm going to write it. I'm going to take this step into account now. You should always write the capital letter first. So I've got big G, little g, big G, little g. Okay, so grey is dominant, capital G. So if grey is present, it's going to be grey. And I've got 100% again, grey here. And I've got 0% white, because for white, I need two little g's. Hopefully this makes sense. If it doesn't, again, I'd recommend go and watch other videos because people can explain it a lot better than me, maybe. Okay, probably. Let's press pause now, see if you get it. If you don't get it, then go back and try and watch other videos, but maybe you're just getting it, okay? It's really hard to tell without you being there, which is why it's hard to do these videos, because I have no idea. There's no feedback coming, so please leave comments. And I don't know if I'm going too fast or maybe I'm just going too slow and you're like, oh, come on, get on with it. Anyway, press pause, see how we're going to do. Okay, let's see how we did. Big G, little g, big G, little g, big G, little g there. Little g, little g, little g, little g. That's the answers I got. So I've got a 50% now. So there's only 2 out of 4, which is half, is going to display that dominant grey. And then I've got 2 here, 2 out of 4, that's both recessive. So when you cross a heterozygous female with a homozygous recessive, so this was a very similar question to the last one we did, but instead of dominant now, the male is recessive, and you can see just how much impact that has. So there's a good chance, well, it's a 50-50 chance, that it'll either be grey or white this time. So and that's how I would write that answer. Again, we can get, as we go into this now, the answers will get a little bit more complex, because I could say that 50% there are heterozygous, and I could say 50% are homozygous recessive. So I can go into a little bit more detail, especially when you're doing this for an exam question, a 6-mark exam question, you'd be expected to use these key terms, which is why at key stage 3, we're just going to leave it there, and then key stage 4, we're going to start pushing this a little. Now we've got this from BBC Bite Size. I'll read it, but we're doing most of this anyway. So this is how, because at key stage 4 you would be expected to construct your own squares. So the first thing you do is to determine parental genotypes. The genotypes, the letters that we use to represent these alleles. And we've kind of been doing that, okay? You get most of that from the question anyway. So you got whether it's a big A or a little a, big B or a little b. This bit we haven't been doing. Step 2, split the alleles for each parent and put them on the edges of the Punnett square. That's the only bit we haven't been doing, because I've been doing that for you there. All right? Number 3, work out the new possible genetic combinations. We've been doing that, okay? And then number 4, provide in your answer the probability or the ratio to determine the phenotype, the characteristic displayed. We've been doing that. So there's only step 2 we've got to do, really. And we're going to practice that now. Now, key stage 3, I would advise, if you're year 7, definitely year 7 and 8, just go for it. Just because, you do anyway. So just challenge yourself, because I reckon you can get it. The inheritance of fur colour in mice is controlled by a single gene. Again, there it is, a single gene. White fur is recessive, A. So I'm just going to go white is recessive, little a. And grey is dominant, big A. tells me that in the question. Sometimes I'd have to make these letters up. Now, two mice that are heterozygous, so they're both different, so they've both got big A, little a, they're crossed. Okay? Determine the probability the offspring will have grey fur. So let's answer the question once we've done the square. So just like noughts and crosses, I'm going to do two lines, two verticals, two horizontals. Now I'm going to bring one letter down, and one letter down to there. Again, I've got a female now, she's heterozygous as well. I'm going to bring one letter there, and one letter there. So I've got big A, little a, big A, little a. Now I need to cross these, just as I've been crossing them. That's the only bit that I haven't done so far. So I've got my big A, little a. So when I cross these now, big A, big A, big A, little a, get my big A, little a there, and then little a, because I've got little a there. And now, I've completed my genetic cross. I can interpret my genetic cross now. So, 25% is homozygous dominant, 25% is heterozygous, homozygous recessive, and 50% is heterozygous. What's the question asking me? Determine the probability the offspring will have grey fur. Well, grey is the dominant gene, so I'm looking for that capital A. I got 1, 2, 3 out of 4, so there's a 3/4 of a chance, so I can say 75% chance it's going to be grey. Yeah? Let's try another one. Okay, the hardest bit is reading the question, because the questions themselves are a bit of a, Coat type in dogs is determined by a single gene. There's that thing again, it's determined by a single gene with two alleles. The straight coat allele is dominant, and the curly coat is recessive. Now it hasn't given me letters this time, so I got straight, and we got curly. I haven't got letters, they haven't given me letters. So let's make letters up. And again, I'm not going to pick letters like C or S, because it's really hard to know which one is capital S and lowercase s. I'm going to pick two letters that I can really know the difference. So I'm going to pick R. Could have been E, could have been anything. R, little r. Because the straight one is the dominant, so that's the capital. The curly one is the recessive, that's the lowercase. Now, let's see what combinations I got. I've got a homozygous dominant male. So I'm going to put him up the top. He's homozygous dominant, so he's R, two capital letters, homozygous the same. And I've got a heterozygous female. I've got a R, little r. Heterozygous different. Let's do my cross. Okay, so I'm just going to bring these a bit clearer now. R. So I cross these two, R. I cross these two, R. I cross these two, R. And I cross these two, R. What's the question asking me? Determine the probability the offspring will have a straight coat. So, well, I can already see there's 100% probability or chance that they're going to have a straight coat because they've got a capital R in each of them. Okay, there's no recessives here. And to break it down further, I've got 50% homozygous dominant and I've got 50% heterozygous there. So the answer to my question is a 50% chance. Okay, they're going to display straight. You have a go now. Press pause. See how we get on. How did we do? So the first thing we needed to do is to interpret the question, really. It's not giving us letters again. So if I just zoom in, it says flower, genetically, the white allele is recessive. So I've said white is lowercase e. You've more than likely got different letters to me now. Could have used R again, but I've just used E this time. And then the pink is dominant, so I've said capital E there. I've got a homozygous recessive there, so the same recessive crossed with a heterozygous. And then I've put that into a Punnett square, and I've come out with a 50% chance that it's going to be pink, because I've got two capital letters there. If you're not getting this, I would really recommend going back and watching the examples again, or even going to watch another video. If you type into YouTube, Punnett Square practice or genetic crosses, you're going to get someone showing you exactly what I'm doing. Just be careful. You might get a dihybrid cross. You want monohybrid is the word. Let me just, monohybrid is the word you'd need to put in but they're the simplest crosses you can get, or single gene crosses, okay? Now we're going to do a quick quiz before we go on to the assignment, just to check that we're all there, and then I'm going to dish out the assignment. So press pause. Question 4 is a bit of a tough one, but have a think about it. Where would you find a gene that is on a chromosome, or you could have said DNA? Okay, you find a gene on the chromosome. What does the term homozygous mean? It's the same, or you could have gone into more detail and said the same genotype. It's a more specific answer there. What type of letter would you use to represent the dominant allele? It would be a capital letter. And then what does a Punnett square show? That was a tough one. It's possible genotype outcomes for offspring. Okay, tough one that. Let's have a look at the assignment. So the assignment, complete it on a piece of paper and photograph it is the easiest way. Some of you have been using the PowerPoint, so we'll upload the PowerPoint as well. And I've left plenty of space that you can fill it in if you want to. But I think these crosses would be quite difficult to do on PowerPoint. So a piece of paper is fine, and you only need to do really the questions that I set you now, and I'll set you those. Use the layout that I've shown, as much information as you can give me. So even talk about the terms homozygous, heterozygous, what percentages I've got, things like that. Okay. Now there's 8 questions. There's 3 for Key Stage 3 and there's 3 for Key Stage 4. And then you've got 2 challenge ones. What I would like though is for everyone to have a go at them all. Now you don't need to do that in order to pass the assignment, but especially if you're year 7 and year 8, I would be really looking to grab this by 2 horns, okay? Year 7 and 8 to complete 1 to 3. So I'll be providing the crosses already laid out for you, just in case you're struggling with this topic. But then questions 4 to 6, you need to draw your own Punnett squares, and then your 2 challenge questions are a little bit more difficult, more sort of edging towards the A level. Oh no, it was sort of higher tier. I wouldn't even say it's A level, it's higher tier. I'm not going to be doing the first one like I've done before, the first problem with you, because I think there's enough examples in this that I've done with you. So I'm going to show you the questions, and then you just submit them, okay? So your first question is there. So the height of a pea plant is controlled by a single gene, which has 2 alleles, tall and short. Again, these are all versions of the same question we've been doing. I'm not going to read each question. You can just flick through these, press pause, read the question, zoom in, do what you need. Again, if you're watching this on a phone, I would say the quality is going to be pretty bad. So try and get this at least on a computer, or sometimes on your TV, you can get this zoomed in. So I'm going to go to question 2 now, which is down here. Or download the PowerPoints, and then you'll see that you can spend as much time on these questions as you want now. Here's question 2. Question 3. Question 3 there, you can see I haven't put the letters. I've given you the square, but I haven't put the letters in, so tougher there, okay. Question 4. This is first Key Stage 4 one. Come up with your own cross. Question 5. Question 6. Challenge 1. And then challenge 2. Okay, hopefully you've all got something from this video, and you know, I think it's going to be a while before we're back in school, especially looking at this this situation at the moment. It's probably likely to be September, so we're going to keep doing this throughout this term, and at least we can try and keep on top of things then. Thanks for watching. Give it a thumbs up if you can, although it means nothing really, so don't bother.